In the world of drug discovery, the end goal is to produce a synthetic drug. Synthetic drugs are made or discovered to inhibit protein activity or enhance other related pathways. The process of drug discovery involves first identifying the problem, second, studying its physiology, and third, selecting a target, which is one of the most critical parts of drug discovery. And after we've selected this target, we need to either design a compound using rationale design or screen a library using techniques like high-throughput screening. With the drugs that have potential, we test them in vitro and finally choose a lead compound, which will eventually make its way to clinical trials. This entire process is a long journey. It takes at least 10 years to develop a new medication from drug discovery all the way to FDA approval, and often requires on average $2.6 billion. The process begins with in vitro studies in efforts to identify a lead compound, following animal testing to identify properties like efficacy, selectivity, and understanding the drug mechanism and test for safety. Finally, we'll go into clinical testing, which involves 3 main phases and addresses specific questions while also testing its safety. And after this, you apply for an NDA or a new drug application that will be approved and finally go into the marketing phase. Let's take a step back and go all the way back to the beginning, making the drug. If we opt for the rational design pathway, we're basically making a molecule from scratch. First, a target structure needs to be identified. After this, a custom-made molecule can be made using the target as a binding template. We also need to consider the type of drug that we want. A reversible inhibitor, perhaps? These types of drugs bind to a target or a protein for a short period of time. Or maybe it's a suicide inhibitor, which binds irreversibly to a target, creating a permanent effect. And finally, you'd need to choose the drug structure, like size, composition, acid-base properties, etc. When it comes to rational design, there are usually 3 main sources of inspiration of creating your drug compound. 1. Natural products that are isolated from bacteria, animals, plants. 2. Endogenous molecules, for example, ATP or other protein binding partners. Or 3. Design de novo, meaning using your imagination and knowledge of the protein interactions that you need to make. Some tools that are typically used include X-ray crystallography, computational docking software, NMR, and others. And just some other considerations when making drugs include number 1, oral bioavailability. You want your drug to be taken as a pill. That's the best marketed type of drug. 2, you want it to be stable against metabolism and degradation. Since you're consuming a drug, you need it to be able to withstand our digestive system and the enzymes that are involved in it. Number 3, you want it to be small in size, which makes drugs easier to make. And number 4, cell permeable, so that it can be uptaken by cells. On the flip side, instead of rational design, you could also use high-throughput screening, or HTS, to make drugs instead. This is where you take thousands of molecules whose functions are unknown and see if they exhibit biological effects. The pros to using this technique is that you find out the effects of drugs really quickly because you're looking at thousands of compounds at the same time. You could do a cell-based screening as well to directly observe a phenotype that a drug causes, for example, cell death. And this allows you to discard very large numbers of potential molecules. But the cons are that it is very expensive, and also you might not know the target from the start, so you might have to work backwards to figure out the target after you have a hit molecule. So after the disease, target, and hit compounds have been identified, we move on to drug screening. Drug screening involves the evaluation of hit compounds using a bunch of experiments to confirm its effect and safety. It's conducted in the following order: in vitro, ex vitro, and in vitro, which are both artificial environments, in vivo, which is a living environment, and finally, clinical trials. Some considerations for drug screening include, number 1, the effect on cells. Is the drug classified as an agonist or an antagonist? Number 2, looking at isolated tissues. Can you characterize activity and selectivity of the molecule? Number 3, in whole animal studies, you want to observe the effect on interacting organ systems and on the disease itself. And number 4, in a disease model, so the animal with the illness of interest, and you want to see proof of concept. After certain types of tests in drug screening, you might need to make chemical modifications to the drug and adjust dosage based on the results. After drug screening has occurred, the hope is that we found a lead compound. With this lead compound, we move into safety and toxicity testing. This is where the estimate risk associated with drug exposure is tested and observed, considering the use and duration of use. So now we have to consider how much of the drug should we give to a patient and for how long. So in these tests, we want to identify potential toxicity of drugs, design more tests to assess these toxic effects, and predict toxicities that should be monitored more closely during clinical trials. This involves obtaining quantitative measurements like the no effect dose. So this is the maximum drug dose where toxic effects are not seen. The minimum lethal dose. This is the smallest dose inducing lethal effects in experimental animals. And finally, the median lethal dose. This is the dose that kills 50% of animals in studies. These tests help determine the initial dose that should be used for humans in clinical trials. After about 2 to 6 years of these toxicity testings, we finally move on to clinical trials. Clinical trials are broken into 3 main phases, followed by 2 post-marketing phases. In phase 1, we test ranges of different doses in healthy volunteers of 20 to 100 to determine the limits of safe clinical dosage. The predictable toxicities will be detected here, and a series of pharmacokinetic measurements will be taken, like drug absorption, half-life, and the metabolism of the drug. After this phase has passed, we move on to phase 2. We test 100 to 200 patients with the target disease to see if there is proof of concept. A single blind design is done, and a broader range of toxicities will be detected. This phase is the highest rate of drug failures. Only about 25% pass to phase 3. But when we do eventually get to phase 3, we test in more about 1,000 patients to establish and confirm the safety and efficacy. In this phase, the drug is also given as it's intended for the market, so usually in the form of a pill. When the drug is finally in the market, we move on to phase 4, where we monitor drug safety in a larger number of patients, over 100,000, and on average, this number is about 500,000. And we detect for low incidence of rare drug effects. And then in phase 5, there's translational research that can be conducted for other studies using the drug. So now you know all the details about the process of drug discovery. While we are in the midst of a global pandemic with COVID-19, a common question that arises is, when will the vaccine be available? And the answer is, as you now know, not so simple. Of course, for the purposes of the pandemic, this entire process is a lot more accelerated, but still must undergo the same amount of testing as a regular drug would.